All right, welcome to lab number 12 for our course. This lab is going to focus on the ability of different kinds of bacteria to utilize a diverse array of nutrients in their environment. So this lecture is focused on lab 5.7, which is the citrate utilization lab, lab 5.8, which is the decarboxylase test, and finally lab 5.15, which is the urease test. So before we talk about these tests in particular, let's just kind of take a step back and think again about big picture here. When we think about animals that we can see, and here's two examples, a crow and a hummingbird, we think about how they're able to eat and what tools they have that allow that diversity in their diet. So if you think about a crow, the thing that everyone knows about crows is that they will literally eat anything. They eat meat, they eat vegetables, they eat garbage, they eat pretty much, if they can get it down their gullet, they're going to eat it, right? They're generalists. The ecological term for that is a generalist. And as such, they have a particular kind of tool in their beak, right? You think about a beak as kind of like a tool for a bird, and that beak allows them to be very adaptable in terms of what they're able to utilize from their environment. On the other hand, we have a hummingbird, which has a very specialized beak, and hummingbirds can only eat nectar, right? They can only take the liquid sugar, kind of sugary nectar from flowers. And in fact, some hummingbirds that are in Central America and parts of the Amazon have such specialized beaks that not only do they have to have nectar, they have to have nectar from a very particular kind of flower or plant. They're very specialized in what they do. And we can take that analogy and kind of carry it over to bacteria, because bacteria have tools also, right? They of course don't have beaks like birds do, but what they do have are enzymes. They have enzymes that give them metabolic pathways that other bacteria might not have. And that allows them to survive in environments, then use nutrient sources that other bacteria might not be able to. So in other words, the bacteria that have the broadest metabolic pathways, in other words, the most enzymes that are capable of utilizing the most diverse array of nutrients, are the ones that are most likely to survive. They're the least fastidious, and they're able to kind of make do with what they got, right? That's why crows are so prevalent in urban environments, because they can make do with what they have, right? They're going to make a meal out of pretty much anything. All right, so let's carry that concept over to the first of our tests. Our first test is citrate utilization. So here we're looking for the ability of bacteria to utilize citrate. So citrate is a carbohydrate that can be found in the environment. And it's important to point out that this particular test has nothing to do with the citric acid cycle. Remember, the citric acid cycle, or the Krebs cycle, is one step of the central metabolic pathways, and it leads to it's involved in respiration pathways, and that's not what we're talking about. In fact, bacteria that do this undergo a fermentation pathway. So bacteria that can utilize citrate are able to utilize citrate as its sole carbon source. In other words, there's no other sources of carbon around, and if it can't utilize citrate, of course, it's going to die. So so here's the pathway, the citrate chemistry here. This is figure 5.25 of your lab manual. I'll talk a little bit more about this in the next slide. In fact, I probably should put this on the next slide, because I want to talk about the recipe first. The Simmons citrate agar is one of the few agars that we have that is a defined medium. And remember that defined media we can trace the molecular amounts of each component of the bacteria, or of the recipe. In other words, we can calculate the molar quantities of every single part of that recipe. And that's important because we want to make sure that the only carbon that's in that media comes from citrate. And we also want to make sure that the only source of nitrogen is coming from ammonium phosphate. In other words, we can't have peptone, we can't have digestive casein, we can't have beef extract, and so on, because all of those typical nutrient sources have a very diverse array of carbon and nitrogen sources. And we want to make sure that bacteria that are in this media can only use these two sources. Next, we have an indicator. Remember that all of these differential tests have some type of indicator either already in the media or we add it after. This is one of the media that it's already contained in the media when we inoculate, and the indicator is a pH indicator called bromothymol blue. It turns out really pretty. This is probably my second favorite, maybe next to MacConkey agar in terms of prettiness. It turns this really beautiful blue color above pH 7.6. All right, so what is actually happening here in terms of the metabolism? So in order for this bacteria to utilize citrate, it has to, what we're really testing for is the presence of these two enzymes, citrate lyase and citrate permease. And let's go back. So let's talk about these two enzymes in the context of that flow chart on figure 525. Here's citrate lyase here, and then here's citrate permease here. So what this bacteria is able to do is they take citrate, and using citrate lyase, they convert citrate to oxaloacetate. And then oxaloacetate is imported into the cell at this point using the enzyme citrate permease. This import changes oxaloacetate into pyruvate. And of course, we all know that pyruvate is that end product of glycolysis. So now we have pyruvate inside the bacteria. And now this bacteria can ferment pyruvate to produce ATP. Now, that should raise a big alarm in your head because that's not what we ever talked about. If you think about what we talked about with pyruvate, it's an end product of glycolysis. And then it usually it accepts what electrons from our electron carriers to turn into an acid, an alcohol, or a gas. So this is kind of one of those little tiny little white lies that I told you in the metabolism unit, is that some bacteria can actually have further fermentative pathways that produce energy. So the bacteria that are citrate positive are an example of those bacteria. So they can actually take pyruvate, convert it into other end products, such as acetate, lactate, acetoin, and so on. And that pathway, in the case of acetate anyway, right here, produces energy for the cell. Now, crazy small amount of energy, right? Super tiny. Remember how bad fermentation is in producing energy? Well, this is even worse, right? So it's a very, very low energy yielding process. But again, it's going to save save this, I must be hungry. It's going to save this bacteria's life if the only source of carbon is that's laying around and is able to be utilized is citrate. So in order to be citrate positive, in other words, in order to have this pretty blue color on this test, this bacteria have to meet a lot of standards. It has to have both of those enzymes, the citrate lyase, the citrate permease. It has to be able to ferment that pyruvate to produce ATP. Remember, if a bacteria doesn't produce energy, it dies. And so this is the only pathway that it has. So it has to do that. And then it also has to be able to utilize ammonium phosphate as its sole nitrogen source. Because remember, if it can't incorporate nitrogen into its DNA and some of its amino acids, it's going to die also. So all four of these conditions must be met in order for bacteria to turn that media blue. So let's look at what happens. Like if we kind of think about this in terms of a flow chart, if a bacteria can utilize citrate, then therefore it can survive, and therefore it's utilizing the ammonium. That ammonium utilization increases the pH. So this is one of the few, have we talked about one yet? I think this is one of the few bacteria differential tests that we're actually looking for an increase in pH. The next one that we talk about, the decarboxylase test, does this as well. But here we're actually looking for an alkaline pH, something above, I think it's 7.6, will turn that media blue. And that's a positive result for this. And again, why do we really care about this? What's the application? This is one of the hallmark characteristics of many important members of that Enterobacteriaceae family. And so it's part of that IMVIC battery of tests. Remember, IMVIC stands for indole methyl red, Vosges-Proskauer, and citrate. So that last test, the C in IMVIC, stands for citrate test. So if we do those tests on our enterics, those potential pathogens, we can get a better idea of what potential bacteria we're working with, identify, and therefore we can determine a treatment regimen. All right, so let's look at some results. So if we were doing this in lab, we would look at 2 bacteria, E. coli and E. aerogenes. You can see, remember, I'm going to try to start including an uninoculated control. These uninoculated controls, we didn't really talk about controls, unfortunately. It was one of the kind of conversations we would have had in person. I did that PowerPoint on it. And remember that uninoculated control in a way is a negative control. It should tell you what, excuse me, it should tell you what no result looks like. It should tell you what no result looks like, and therefore it's a type of negative control. It also tells us that our media is sterile and that we don't have a reaction in the absence of a positive bacteria. And it also tells us that our pH indicator doesn't just kind of spontaneously turn color after incubation at 37 degrees, for example. So it's also a nice color comparison. We can see very clearly that tube number 2 is a negative result because it looks exactly like that uninoculated control. All right, so go ahead and write these results in for your data sheet on lab number 5-7, and let's move on to the decarboxylase tests. All right, so the decarboxylase test, it's a fun test. It's a fun battery of tests because it reminds me a lot of the phenol red tests that we did. If you recall, in the phenol red test, we were looking for the ability of a bacteria to ferment a bunch of different carbs, right? We could run all these different types of carbohydrates in the media, and we would look for acid production or yellow media, and we would say it can ferment glucose, it can ferment lactose, it can ferment sucrose, and so on. And what we're doing here is we're looking for the ability of bacteria to actually utilize amino acids as a nutrient source. And they can do this by decarboxylation of those amino acids. Let's remind ourselves, if you've had biochem or I think you talk about this in Gen Chem, what the basic structure of an amino acid is. We have that central carbon. On one side of our central carbon, we have our amine group. On the other side, we have our carboxylic acid. And then what differentiates amino acids from each other is that R group that comes off of our central carbon. So what happens here is that some bacteria can remove this carboxyl group, and removing this carboxyl group from using a specific enzyme called a decarboxylase will result in an amine. So based on what this amine is, we now have kind of a tiny little Lego, right? So this is a catabolic reaction. We're breaking 2 things in half. The resulting amine can either go into the central metabolic pathways to produce energy for the bacteria, or it can be made into something else using some anabolic pathway. So this is a big advantage for bacteria in that they can take these various amino acids and break them down and turn them either into energy or utilize them for other things. So when we look at the recipe for this particular test, we have lots of nutrient sources. We have peptone, we have beef extract, we have glucose. We have our indicator in the pH indicator bromocresol purple. We also include peroxidol. Peroxidol is just an important coenzyme, for those decarboxylases to work. You can see it right under here. So really this addition to the recipe just helps our decarboxylases work if they're there. And then again, just like our phenol red test, we can only add 1 amino acid at a time. So we want to add only lysine or only arginine. Those are the 2 that we're going to be looking at today. So we want to always keep those in separate tubes and label our tubes with the amino acid that we're looking at. And then another interesting addition to this particular test is the addition of mineral oil. So mineral oil is going to be laid over the top of the tube, and this basically prevents oxygen from getting in the media and promotes a fermentation pathway because we're removing oxygen from the environment. So the timing of this is kind of interesting because what happens with bacteria that can do this, they first remember, what is our cardinal rule with bacteria? They will always eat glucose first, right? So and glucose is present, look at our recipe, it has glucose here. But because we have that mineral oil, they can only ferment glucose. They can't aerobically respire because they don't have oxygen present. So we're inducing that fermentation pathway of glucose. and that's going to lower the pH. So this happens, I'll clarify this later, but let's just say early, okay? And that lowering of the pH is going to turn our pH indicator, bromocresol purple, it's actually going to turn it yellow, this nice, bright, pretty yellow color. And that acidic environment is going to turn on expression of these decarboxylation enzymes. And decarboxylation is going to start processing those amino acids. And then our amines are going to build up in the media, turning the media alkaline and thus purple. So if we have kind of the original color, which is kind of this, I don't know how to describe it. It's kind of like this olive-like color, or a yellow color. Those are both negative. They're both considered negative for decarboxylation. The only positive result that we have is purple. Now, one of the issues with this test is that this can take a really long time, sometimes up to 4 or 5 days for a bacteria to do this, because we're waiting for those amines to build up in the media and collectively turn that pH alkaline from a fairly pH media. So if we read these results too early, we run the risk of reading this as a false negative. So you want to be very careful anytime you do a decarboxylation test to look at what the literature says to wait the incubation time on these. For example, I think the lysine and arginine decarboxylase test that we would have done in lab take a minimum of 96 hours to turn purple. So we want to make sure that we're kind of taking our time and giving that bacteria plenty of time to decarboxylate those amino acids. So let's look at the 2 bacteria that we would have done in lab, and we would characterize them based on 2 amino acids. We have a 96-hour incubation time, and we're going to have an uninoculated control. We're also going to have a base broth that has no amino acids in it. This is letting us know that the media can't turn purple in the absence of those amino acids. And of course, our uninoculated control is a negative control to let us know what no result looks like, that our broth is sterile, and so on. So we can see here that we have 2 bacteria, Enterobacter aerogenes and Enterobacter? Oh, that might be Escherichia. I forget what the genus name of Cloacae is. But we're going to look at these two bacteria in regards to their ability to decarboxylate the amino acid lysine and the amino acid arginine. So let's go ahead and you go ahead and record these results as if you were doing this in class. And then when you're done with that, think about what would happen if we forgot the mineral oil. If we forgot the mineral oil, could we trust these results? Well, no, we couldn't, because absent of mineral oil, we could have respiration processes occurring, and then we don't really know if the purple color is due to decarboxylation or if the yellow color is due to accumulation of some other acid, and so on. So this is something students do all the time when they're doing their unknowns, is they forget to add the mineral oil to their decarboxylase broths, and then they have to repeat them after waiting like a week to get their results. So big pain in the butt for students. All right, let's look at some variations on this. So if you were to, well, first of all, let me just kind of show you what they look like in real life. Here's a picture I stole off the internet of some bacteria. I've never even heard of this bacteria, K. oxytoca. No idea what this is, but we see it in a base broth, and then different types of amino acid, and you can see that the only amino acid that this particular bacteria is capable of decarboxylating in this case is lysine. And so you can see a very clear purple result versus yellow for the other bacteria. Now, here's a kind of challenge question for you. What if you had bacteria X, bacteria Y, you had an uninoculated control, and you also had inoculated these 2 bacteria into a base broth as another type of negative control. And after 24 hours, you see yellow for lysine for bacteria X, and you see purple for lysine for bacteria Y. How would you read these results? What would you do if you were in lab and you pulled these tubes out of the incubator after 24 hours and you saw that X was yellow and Y was purple? How would you record that? So something to think about. Probably going to be a practical question. All right, so let's do our last one, and this is urea hydrolysis. So very specific bacteria, as you can imagine, many bacteria that are pathogens of the urogenital tract can do this. Also, interestingly, a bacteria called Helicobacter pylori can do this as well. That is the causative agent of stomach ulcers. Helicobacter pylori is one of the rare bacteria that can actually survive in our stomach and cause gastric ulcers. And it does this by utilizing urea to neutralize the stomach acid in its immediate environment. It's kind of a cool little bacteria. And also the guy that discovered it won the Nobel Prize for proving it. Up until that point, people thought that gastric ulcers were caused by stress and not by a bacteria. So what does this test do? Well, it tests for the ability of a bacteria to hydrolyze urea into ammonia. And so again, what we're always really testing for is that enzyme. And the enzyme in this case that can make this reaction happen is the enzyme urease. The recipe here is almost, it's almost a defined media. It has this tiny little amount of yeast extract, a trace amount of yeast extract in the media. It has a buffer to prevent quick changes in pH. And then, of course, it has our substrate, urea, and our indicator in phenol red. If you remember, we used phenol red in our fermentation test, but in that test, we were looking for it to turn yellow. We wanted it to turn acidic and a yellow color. In this test, we're actually looking for it to go into an alkaline. Oh, wow, all three of these, actually. So all three tests that we did today, the citrate, decarb, and urea, have an alkaline result to them. I never really thought of that before. But when the pH goes above, I don't know what the exact number is, but into an alkaline environment, that phenol red indicator is going to turn this really pretty hot pink color, like you see in tube B. So what happens here is that urea, urea is present in again, think about a bacteria that's hanging out in our urogenital tract. It needs the most available nutrient resource, and one easy source of nitrogen is urea. And it can take urea and convert urea into ammonium. Let me just double check. Yeah, it can convert it to ammonia and carbon dioxide, and then it can utilize that ammonia and it can incorporate it into some anabolic pathways to build precursor metabolites. The interesting thing about this test is that it's a rapid test. You can read these results, and you actually want to read these results in less than 24 hours. And sometimes if students, when students were working in the lab, they would inoculate at the beginning of lab, and by the time they left, it was already pink. So you can sometimes get results in just a couple of hours. And the value of this test really is to differentiate Proteus species, so Proteus vulgaris, Proteus mirabilis, pathogens of the urogenital tract from the other enterics. So most enterics don't have this ability to process urea, but these Proteus species do. So we have our, so we have our controls. If we were doing this in the lab, we would have our uninoculated control. Here we can see our Proteus species, Proteus vulgaris, is turning that media a beautiful, pretty pink color. And we have our negative control in E. coli, which is unable to turn that media pink. And in fact, I would argue that E. coli probably can't even grow here. And that's because if it can't look at the recipe. If it can't utilize urea, what is it gonna eat? Right? Even E. coli, E. coli is a very, very resourceful bacteria. It can pretty much make anything out of anything. It's kind of like the crow of bacteria. But even E. coli probably couldn't grow in this media because it simply doesn't have anything to eat. The yeast extract is so small that it really isn't enough to support growth in this media. So in a way, you could almost say that this is I don't want to call it a selective media, but it's almost like an enrichment media maybe, because only bacteria that can utilize, like I would consider the citrate test and the urea test enrichment tests, and or enrichment broths or enrichment media, because only bacteria that, like, growth is a positive result. If you think, right, if you look at the citrate test, sorry, you thought you were done, didn't you? Sorry. So if you think about the recipe, where where did it go here? There in citrate, the only source of carbon is citrate. So if this bacteria can't utilize citrate, it's not gonna grow. So really, growth is a positive result. And if we see growth, we should see that blue color. I guess that you could have some. I think I'm gonna sneeze. Sorry. Nope. Maybe. Oh my goodness, I'm sorry. So if we see growth, we should see that alkaline media color, because the fact that it can grow means that it's converting that ammonia into some alkaline product. So citrate does this, and urea does this too, right? If it can grow, if it can utilize urea as a nutrient source, as a source of nitrogen, then it can grow in this broth. Otherwise, if it can't, it's probably not even going to grow. All right, that was a quick one. Almost under 25 minutes for this lab. Hopefully they keep getting shorter and shorter for you as we go along. All right, that does it for this week.